Hello my dear students, welcome to our first video lesson in microbiology and parasitology. In this video lesson, let us talk about the growth of microbes, particularly bacteria. Now, just like any other living organism, a bacterial cell also possesses this property which is called growth and development. Okay, so this is really the nature of any living cell. Just like eukaryotic cells, which divide through the process called mitosis, bacteria also multiply, but in a different manner, of course. Bacteria divide by means of this process called binary fission, in which one parent cell divides to form two progeny cells. Okay, but of course, it's the number of cells produced per single cell is the same also with mitosis. The only difference is that the process in bacteria is not called mitosis, but it's binary fission, wherein the single cell here will divide to form or to produce this what we call two progeny cells. But of course, the DNA here, which is contained in the chromosome of the bacterial cell, will also double or will also duplicate, in order for each of these cells to have its own exact copy of the DNA as the parent cell. Okay? Now, because one cell gives rise to two progeny cells, bacteria are said to undergo exponential growth in just a matter of hours. Let's take into consideration this table here. Okay, so this is the the number of cells. This is the exponential growth, wherein one cell will undergo binary fission to become two daughter cells. And of course, after 4 generations, the single bacterial cell will become 16. And that happens for only a few hours or less than an hour. Okay? Now, let us proceed. So, in this picture here, you can see that the single bacterial cell doubles, and after a few minutes, it continued to increase and increase and increase until it reaches the 4th generation, wherein there are already 16 bacterial cells. So this is just an example, an illustration of the table that is shown above. Now, the doubling time of bacteria, particularly the E. coli or Escherichia coli, is as shorter as 20 minutes, So, E. coli is a kind of bacteria, particularly a gram-negative bacteria. It is also rod-shaped that usually inhabits our large intestine or our colon. That's why you can see them always in fecal materials or in feces, or dumi ng tao or hayop. Now, in the case of Mycobacterium tuberculosis, its doubling time is 18 hours, in maximum. Means to say, doubling time means to say the time in which the number of these bacterial cells will double in number. Okay, by the way, Mycobacterium tuberculosis is another type of bacteria, particularly a member of the Bacillus family, and this one is the causative agent of the notorious agent of a disease called tuberculosis. Okay, imagine how fast its doubling time is. Now, the growth cycle of bacteria has 4 major phases: the log phase, or the lag phase, the log phase or the algorithm phase, and of course this log phase is also known as the exponential phase. The third one is this what we call stationary phase or the steady state, and the last phase is of course the death phase. Okay, so let us dig deeper into this 4 growth cycle of bacteria. Well, the first phase is called the log phase. As you can see in this line graph here, the log phase is the first phase of the growth cycle wherein there is no increase in the number of living bacterial cells. Okay, no increase yet. But what's happening in here is that the bacterial cell gathers metabolic nutrients that are being used or beneficial to continue its growth cycle. Okay, so there is no growth or there's no division or increase in the number of cells here during the log phase. So this one here is the log phase. After a few minutes, this the bacterial cells will divide exponentially. Therefore, as you can see here, the line graph is also increasing abruptly. Okay, so the log phase is also known as the exponential phase or the exponential increase, sudden increase in the number of living bacterial cells. Okay, now after the log phase class, you can see here. Okay, so again, this is the portion here is called the log phase or the exponential increase in the number of bacterial living cells. Now in this phase, there is this what we call rapid cell division. Okay, so antibiotics such as penicillin are more efficacious during this time. Okay, so parang onset of the occurrence of disease. Okay, so that's the time wherein most antibiotics are more effective because this time, okay, so this time there is this what we call biosynthetic processes that are occurring. So it is more appropriate, or this is the appropriate time to disrupt their biosynthetic processes. Okay, so this is the log phase wherein there is sudden increase in the number of bacterial living cells. Okay, let's proceed to the next phase, which is called the stationary phase. Okay, so after the log phase or exponential phase, there is this what we call plateauing. Okay, as it is shown in the line graph here, meaning to say there is depletion of nutrients, or might be toxic products are being administered that causes the growth of the bacteria to slow down. Okay, if you can still remember in our previous slide here, okay, during the log phase, this is the most crucial phase in here wherein antibiotics are more effective. Okay, because at this time there is what we call biosynthetic processes that are happening. Okay, so if let's say for example toxic products such as antibiotics are being administered, it will result to this what we call stationary phase wherein there is the reaction of the cells, okay, to toxic products, in which those toxic products may cause the growth of bacteria to slow down. Okay, or might be there is this what we call nutrient depletion. Papaubos na yung nutrients, yung source of energy ng bacterial cell. Okay, so this will happen, or this occurs until number of new cells produced balances the number of cells that die. Okay, and this results to this what we call steady state or the equilibrium state, wherein the number of cells, the number of new cells roughly equals the number of death cells, okay, or the number of the cells that died. Okay, now in the case when bacteria are grown, okay, in special apparatus, of course there are bacteria that are being cultured in the laboratory, which are usually used for researches. The scientists do this, what we call addition of fresh nutrients into the medium, okay, or into the chemostat. This is what we call the special apparatus wherein cells, bacterial cells, are grown. Okay, so what's the purpose of this? Adding fresh nutrients to the medium and removing the waste products constantly. Okay, the result is the bacterial cell will remain in the exponential phase or in the log phase and will not enter the stationary phase. So what is the implication of this? Okay, the implication of this is that the growth of bacteria will be maintained, okay, and no bacterial cell will die. It will just remain in the log phase or in the exponential phase. Okay, so the bottom line there is, if there are plenty of nutrients or plenty of energy sources, the bacteria will continue to divide and multiply. Okay, so in order to kill the bacteria, one of the major or one of the techniques is to starve them. Okay, so just like any other living organism, bacteria need nutrients, a source of their energy for them to endure their existence. Okay, now after the stationary phase, there is this what we call the death phase. Okay, so during this phase, class, there is this what we call exponential decrease in the number of living cells. Okay, so there is the decline in the number of viable cells. So that's the four phases of the growth cycle of bacteria. Now let us proceed to this another topic, which is called the obligate intracellular bacteria. Okay, so we will discuss this in our next video lesson.